In Chapter 8, we turn our discussion of genetic material to chromosomes and cell division. So every time a cell is going to pass on its genetic information, it has to divide. There's different types of cell division. We'll talk about those, and there's many different reasons for cell division. But the ultimate goal of all cell division is to pass on genetic information and to pass on that genetic information faithfully to the next generation of cells. So in Chapter 8, we're going to talk about a little bit about cancer, which is uncontrolled cell division. We're going to talk about the different types of cell division. There are two types of cell division. We're going to talk very, very briefly about DNA replication and how the cell prepares its genetic material for cell division. Then we're also going to talk about different types of chromosomes. We'll talk about our normal chromosomes and our sex chromosomes, those chromosomes that determine the sex of an individual. And then we will also talk about chromosome abnormalities and some of the genetic disorders that arise from different types of chromosome problems. So our chapter starts off with talking about a special structure called a telomere. So a telomere is a section of non-coding repetitive DNA at the end of each of our eukaryotic chromosomes. And remember that eukaryotic chromosomes are linear, so at each end of the chromosome there's a protective cap called a telomere. So let's back up a little bit and talk about why telomeres are there. So cell division is an ongoing process that occurs in most organisms. It's the process by which cells divide for growth or for development, for replacement of old cells. And if that process of cell division is disrupted, there can be some very serious consequences for the organism. So in eukaryotic cells, telomeres act as sort of a protective cap during cell division. So every time cell division happens, DNA replication occurs, and the chromosomes are copied and divided up and passed on to the next generation. But telomeres are needed because at the end of DNA replication, as the replication enzymes get all the way to the end of the chromosomes, they actually leave a little bit off. Now, the reason for this is a really complicated molecular biology process, but basically those enzymes that are performing DNA replication can't make it all the way to the end. They can't make the very tips of the chromosomes double-stranded. And so the ends of the chromosomes are actually lost. So telomeres sort of act like an odometer. So the odometer is the mileage ticker in your car that just keeps track of how far your car has driven. So telomeres sort of act as a ticker for keeping track of how many times your cell has divided. Now, telomeres do this because ends of the chromosomes are lost during replication. So every time the cell divides, it loses a little bit of telomere. Once the telomeres are gone, the cell either dies or no longer enters cell division. And this is sort of a protective mechanism, because if you continue to lose genetic information and the cell still survives, you're eventually going to get past the telomeres and get into important coding DNA. Now, if you actually do get into important coding DNA, your cell's probably not going to divide anyway. That cell's going to divide and it's not going to propagate its compromised genetic material. So on the flip side of this, there are actually some cells that can rebuild their telomeres. Those cells that are producing sperm and eggs, they're dividing a whole bunch, they can actually rebuild their telomeres because they need to continue to produce whole pieces of DNA. During early development, when all of our cells are growing rapidly and our bodies are growing rapidly, a lot of our cells can reproduce the telomeres, but then they stop after a while. So a baby starts with enough telomeres for 80 to 90 cell divisions in most of the cells. By the time you're 70, you only have enough telomere left for 20 or 30 cell divisions. And so once your telomeres are actually lost, that cell will stop dividing and die. telomeres are also really important in disease. There is a particular disease called Hutchinson-Gilford progeria, and individuals affected by Hutchinson-Gilford appear to age much more quickly than other individuals of their age. And they believe that this is related to telomere loss. So these children are born with shorter than normal telomeres, and because of that, they appear to age rapidly. So they acquire these characteristics. They're born normally, and then they acquire these characteristics that make them appear much older. They have the same characteristics of individuals who are much, much older. Another problem that we can get from telomere that comes out of a sort of defective telomere mechanism is cancer. So a lot of cancers actually gain or maintain the ability to rebuild their telomeres. Now, if you lose your telomeres, cell division stops. But cancer is uncontrolled cell division. And the reason for many, many cancers, or the reason that these cancer cells continue to divide and to produce tumors, is that there's telomere rebuilding occurring. And so when normal cell division, even uncontrolled cell division, were to occur, it would eventually stop because telomeres are being blocked, or telomeres are blocking cell division. But when telomeres are continuously rebuilt, it basically means that that cell line can live on forever. And this is one of the reasons that makes cancer so awful. So we're going to come back to cancer in just a few minutes when we talk about cell division. But for a minute, we're going to briefly review the structure of chromosomes. So prokaryotic and eukaryotic chromosomes are very different. Prokaryotic chromosomes, or prokaryotic cells, have circular chromosomes. Now, if you look here, these are twisted all around each other. This is what would happen if you, say, took a rubber band and held one end and twisted the other end, and it would sort of coil up on itself. You get this circular chromosome that is very tightly wound around itself. And packaged sort of really, really neatly into the prokaryotic cell. Eukaryotic chromosomes, on the other hand, are linear. Now, you can't really tell in this picture because it looks like a bunch of spaghetti, but we have one end of our chromosome here. It twists and turns around until we get all the way out to the other end of our chromosome, to our DNA. Now, these chromosomes are also very tightly packed. Think about doing the same thing. If you have a shoelace and you twist it on one end and hold the other and twist it and twist it and twist it, it eventually coils up on itself. And that's sort of how our DNA is packaged into our cells. We've got our DNA structure here with our base pairs and our sugar phosphate backbone. It is, of course, a double helix. Eventually, in order to start the packing process into the cell, it is wrapped around proteins called histones into a very neat little package. And then those histones are sort of packaged around themselves and curled and coiled and twisted nice and tight so that you can get a huge amount of genetic information into a very, very tiny cell. Now, DNA replication is a little bit different depending on which cell you actually are, although the overall process is very much the same. Prokaryotes divide by a process called binary fission. So, cell replication is the whole process of a cell duplicating each of its chromosomes and then dividing the genetic information, the cytoplasm, and even the plasma membrane or cell wall, or both, depending on what you have, dividing them into two daughter cells. So if you're a bacteria, this whole process is quite simple. The parent cell eventually divides into two daughter cells. So here we have our prokaryote. We've got the cell wall, the cell membrane, our circular DNA. Remember, even though it's circular, it's still double-stranded. And this cell is going to begin DNA replication. So if you look here, we've got a new strand forming here and here. Our original double helix has pulled apart, the backbones have separated, and then we get little proteins in here that come through and add complementary information to both sides. This continues until we have 2 full pieces of DNA. Now this DNA is exact copies. It's as if you were to take a piece of paper and photocopy it. It should be exactly the same, carrying exactly the same genetic information. So here we have our cell with 2 chromosomes, identical copies in the cell. The cell begins to sort of stretch out, pinch in the center. The DNA is partitioned into either cell, and eventually we end up with 2 fully formed daughter cells containing exactly the same genetic information that the parent had. So these guys are genetically identical to each other, and they should also be genetically identical to the parent. So this process of binary fission is the process by which eukaryotes divide their DNA, divide their cell, and produce 2 genetically identical daughter cells. Let me go back a little bit. So the process of binary fission is what we call asexual reproduction. That means that one individual produces all of the genetic information for the next generation of the cell. And this is why the daughter cells are genetically identical. There's only one parent contributing information. This will be important when we start talking about sexual reproduction and the process of fertilization. All right, so eukaryotic cell division is a bit more complicated. DNA replication still occurs, the cell divides. But there's a bit more going on, and so we're going to talk about the process of eukaryotic cell division. Now, cell division is only one small part of the entire cell cycle of a eukaryotic organism. So this purple here is what we call interphase. Interphase is where our cells spend most of their time. It's a period of growth and development and preparation for the second phase of our cell cycle, which is mitosis, or the mitotic phase. Now you'll see here that we start with one cell, we go through the cell cycle, we produce 2 cells, and then each of these are going to go through the cell cycle as well. Within interphase, there are several shorter phases. G1 is the first phase once a cell exits mitosis. So this first phase is called G1, and this is sort of a normal growth and development stage. This is where the cells perform their normal functions. This is going to be cellular growth, taking in nutrients, breaking them down, building proteins, expressing their genes, interacting with their environment. So most of their life is actually spent in depending on the cell, most of their life is generally spent in the gap 1 phase. After gap 1, some cells will actually enter a phase called G0. So G0 is sort of a resting phase. G0 is a phase that cells might enter and then never leave, and it just means that they're not dividing anymore. So there are certain cells like our neurons, our heart muscle cells, that never divide. There are others like our liver that might divide very infrequently once growth and development stops. Other cells divide all the time. So most of the cells in our body are not going to enter G0, but there are actually some very critical ones that do enter G0 and then only leave occasionally and pop back into this interphase cycle, or never actually leave. So let's assume our cell has not entered G0. After gap 1, we are going to enter the S phase, or the DNA synthesis phase. This is the phase where the cell really begins preparation for cell division. During the S phase, DNA synthesis occurs. This means that each of the chromosomes are replicated, and we'll talk about the process for that in just a minute. We saw that over there in binary fission a little bit ago. And so each of the chromosomes is going to make a complementary copy of itself, which means that the genetic information in both of those daughter chromosomes should be exactly the same. After that, the cell enters gap 2 phase, which is a second period of growth, and the cell performs its job even though it's got extra genetic information. Usually there's a lot of growth in this phase because the cell's getting ready to split into two. And there's a lot of different proteins and things that are produced getting ready for cell division. At the end of gap 2, we're going to enter mitosis. Now, I'm not going to talk too much about mitosis at the moment because we're going to go into a lot more detail. But in mitosis, the chromosomes are separated, the cytoplasm is split, the plasma membrane is split as well, and the cell, each of those cells becomes their own daughter cell, which is going to enter their own interphase in preparation for the next round of cell division. Each one of these phases of the cell cycle is really tightly regulated by something called the cell cycle control system. Each of those phases is regulated by a checkpoint, basically a gatekeeper at the end of each of these checkpoints that checks to make sure the cell is ready to move on. If the cell's not ready to move on, then one of two things can happen. The cycles can be paused until everything's ready to go, or the cell can undergo basically a programmed cell death. If there's a problem within the cell where the cell is basically not growing and developing normally, the cell can actually die at that point. But each one of these checkpoints is really important. So let me show you where we are. Remember, purple is the interphase, so that's our growth and development phase. Mitosis is our cell division. So let's start here at the end of mitosis. So our new cell enters G1. The first phase, or the first checkpoint after G1 is the G1S checkpoint. So the checkpoint between our gap 1 phase and our DNA synthesis phase. At this point, the cell is going to check and say, hey, is the DNA damaged? Can it be repaired? If not, the cell doesn't want to divide because it doesn't want to pass on faulty genetic information. The cell is also going to ensure that it has sufficient nutrients. Is it healthy and ready to divide? If both of those conditions are met, DNA is okay, sufficient nutrients, the cell's going to enter the DNA synthesis phase and the G2 phase. At the end of G2, we have another checkpoint in which the cell checks to make sure that it's all ready to go for mitosis. Has DNA been replicated properly? Is all the DNA intact? Is the cell ready? If so, the cell enters the process of mitosis. And within mitosis, there is another checkpoint. So this checkpoint's sort of in the middle of mitosis, even though it's drawn at the end. This is called the spindle assembly checkpoint. And it's basically checking to make sure that all the chromosomes have been lined up appropriately and are all attached to special microtubules that are going to pull them apart and separate them during the process of cell division. So once the cell has met any given checkpoint, it is allowed to proceed to the next phase in the cell cycle.